So good evening, everyone. Thank you for making time, and apologies for the delays. So welcome to our fireside chat for this evening. This is going to be our 209th webinar in this series. And today we're going to be taken through a very thought-provoking and interesting topic on transplant immunology that is going to be simplified for the developing world. Our guest speaker for this evening is Dr. Shruti Tapiawala. Myself, I'm Dr. Yuri, and I'll be moderating this session. So just before we kick it off, I want to just introduce Dr. Shruti, and she has quite a rich CV. And as you see, she is a highly qualified nephrologist and a transplant physician with internal medicine training. She also has DNB, nephrology, fellowship from the Indian Society of Nephrology, another fellowship from the Indian Society of Organ Transplant. She is also a fellow of the American Society of Transplant, AST, UHN, Canada, and a fellowship in HLA medicine from Toronto Medical Labs. She is also a consultant. She has had consultant roles at Nanavati Max Super Specialty Hospital, Gleneagles Hospitals, and Anil Clinic in Mumbai. She has held director positions at ATDI Lab, PVT Limited, Precision Transplant in Parel, Mumbai, and Advanced Renal Replacement Therapies in Mumbai. She is a council member with the Indian Society of Organ Transplantation and Women in Transplant in India. Past leadership roles in the Indian Society of Nephrology, West Zone chapter, and also Women in Nephrology India. I know that's just a quick summary, but she has quite a lot of things that she has done in the field of nephrology, and we want to learn quite a lot from her in this presentation. Over to you, Dr. Shruti.
Yes, thank you so much for that introduction, and I'm happy to be here. I have done this talk in the past. I am trying to minimize or simplify what we can do with the transplantation, with what we do in this part of the world. So I will try to take you through what a transplant organ goes through in a human organ. So transplantation, as we know, is the transfer of an organ or a tissue from one part or an individual to another. This was a definition from Dr. Merriam, and this has been the greatest advances of modern medicine to treat end-stage organ diseases. And we have come a long way since the first transplant. If we look at the milestones in transplant, that will explain to you how transplant immunology has developed or how our understanding of transplant immunology has developed. So in 1680s, bone from a dog was used to repair a human skull. So this idea has always been there that if something is not working in a human being, you simply try to replace it. 1881, earliest skin grafts, some using the frog skin. 1930, Karl Landsteiner awarded the Nobel Prize for discovery of ABO blood groups. Another landmark discovery for the mismatched antigen. 1945, Dr. Medever publishes a paper on linking graft rejection and immune system. Around that time in 1952-1954, Dr. Murray did the first successful kidney transplant between identical twins. 1967, first successful heart transplant. 1990, lung transplant. 1992, a baboon liver survives for 2 months in a human being. 1995, you had an AIDS patient receive a bone marrow transplant from a chimpanzee. 2002, first liver transplant between identical twins. And then there has been no stopping. Now we do hand transplants. We have now embarked into xenotransplantation, which is soon going to be a reality coming out of the experimental realm. And there is no stopping. So how do you classify these grafts being transferred from one to another? If you have a graft transplanted from one part of the body to another in the same individual, it is known as autologous. Synergic. Syngenic, it can be transplanted between two genetically identical individuals of the same species. Allogenic, graft transplanted between two genetically different individuals of the same species. And xenogenic, where grafts are transplanted between individuals of different species. And this cartoon shows you that autograft is within the same body, isograft is between two twins, non-identical from the same species as an allograft. Between a different species, it becomes a xenograft, more so ever from an animal to a human being. Now, what are the immune responses to a transplanted tissue? This is very important because we cannot go against our our body, you can say. We have to understand what the body is trying to do in response to something, and we have to support it. So understanding the immune system makes the basis of having a successful transplant, helping the body accept the graft. Transplant rejection is caused by genetic differences between the donor and the recipient, and the differences are between the HLA and the blood group antigens. I will tell you a little bit about HLA in some time. Alloantigens, which vary between members of the same species. So these are antigens which are different within the same species. Alloreaction is a response to the alloantigen, which was different. Alloreactions in the transplantation can be host versus graft or graft versus host. What is that? So when a kidney is transplanted and the recipient's body or the immune system or the T cells attack the transplant, it is host versus graft disease. And when in cases of intestine or bone marrow, when the transplanted organs immune system attacks the host, that is the body of the patient itself, that becomes graft versus host. So this is a very important difference which you should understand, because the bone marrow or the intestine comes with its own immune system from the donor, and that can itself overwhelm the immune system of the recipient and cause a graft versus host rejection. Now, if you look at our immune system, it has innate immunity, which we are all born with, and we have an acquired immunity. So what is innate immunity? It is blood-borne. It has all physical barriers like skin, mucous membranes, saliva, flushing action of the urine and tears and stomach, and this stops the infection before it enters the body. This we are born with. Then in blood, we have the complement cascade, and we have the cells. We have phagocytes. We have neutrophils, macrophages, basophils, eosinophils, natural killer cells, which cause the death of dangerous organisms. And this is also an innate response. We are born with it. So even if there are no doctors in the world, the human system is able to take care of itself. Then there is a direct killing of the bacteria. You know that every time there's an infection, there is a rise in the body temperature, which attempts to kill the organism which is foreign to it. Then you have the complement cascade, alternative pathway, and the classical pathway, which again are going to add to the fuel to the fire when there is a foreign body in the system. This is your innate immunity. Now, what is adaptive immunity? The name itself suggests that you are not born with it, but as time goes by, your body adapts to something which is new. So it can be T cell immunity, it can be B cell immunity, it can be cell mediated with T cell or humoral immunity with B cells. So T cell immunity, whole T cells are released into the suppressor T cells, helper T cells, cytotoxic T cells, and these differentiated T cells, once the T cells are activated, cause the death of body cells that are infected with the virus or otherwise are damaged. B cell immunity is a humoral immunity, which is after an antigen exposure. So that exposure is going to cause lymphoblasts, and then it is going to generate plasma cells, which are going to then release antibodies or make antibodies, and then activate the complement cascade once the antigen is encountered. These lymphoblasts can also form a clone of cells, which can then make memory B cells. And these memory B cells can always release antibodies when that antigen, when and ever that antigen comes to our system. So this chart is very important as the basis of transplant, because when you have HLA antigens from another human being or an animal encountered in your body, you will have a cell-based and a humoral immunity come in. You will have memory response come to those antigens. So if ever the patient has had a transplant in the past or has had blood transfusions in the past, has had pregnancies in the past, that is the time when foreign HLA has been exposed to the immune system. And the immune system has developed an adaptive immunity to those antibodies, saying that this is those antigens, saying that this is not mine. It will make those antibodies and it will have a recognition or a memory response whenever the antigen comes back, which essentially can reject an incoming allograft if it has already been exposed to that antigen earlier. So these are the differences between the innate immune system and the adaptive immune system. If you see the specificities, pattern recognition in innate, and in adaptive immune system, it is the antigen recognition. There is a memory pattern to the adaptive immune system, but not to the innate, because innate is inborn with us. It will remain with us with all the life. It has non-reactivity to cells, which is there in both. So remember this important thing. When you have an HLA antigen of your own, you will not react to it. Sometimes there are antibodies which are formed to your self antigens. That can happen in lupus. They are known as autoantibodies, but they are not pathological for a transplantation. So you will not damage your own self with the autoantibodies. The cellular and the chemical barriers are important in the innate immune system, and in the adaptive immune system, you have lymphocytes in the epithelia, antibodies secreted at the epithelial surfaces. The blood protein cells which are important in innate system are the complements and the phagocytes like neutrophils and lymphocytes. Whereas in adaptive immune system, it is mainly the antibodies and the lymphocytes. So if you look at the innate immunity, this is a cartoon which will give you the difference. So if you look at the innate immunity, you will have a microbe which is coming inside, and you already have a system in place with the barriers and with the phagocytes, and they're immediately within a few hours, they are taken care of and a reaction happens. Whereas in adaptive immunity, what happens is that the antigen has come in. Now the B cells and the T cells are going to try and recognize that antigen, and it is going to try and see if that antibody is already there in the system. The T lymphocytes are going to immediately act by having an antigen and an antibody reaction with a complement adding to the cascade with effector T cells and causing a reaction. Whereas the B cells are going to take time, they are going to take in the antigen, they're going to process it, make antibodies and release them into the system. So that is adaptive immunity for you. Functions of the innate immune system: identification and removal of foreign substances, recruiting immune cells to the sites by cytokines, activation of the complement cascade, and activation of the antigen presentation cascade. Now, if you look at the principal mechanisms of the innate and adaptive immunity, you can see that there has to be an antigen recognition first. After that, there will be a clonal expansion, differentiation, lymphocyte activation, because once you have an antigen antibody response, you need to have a troop of cells with the effector cells going ahead and causing the damage, and it will start making antibodies and effector cells go to that area. There will be a contraction homeostasis. There'll be a memory response wherein you will have some cells go into a memory response, and whichever are active antigen antibody response will cause the cascade. Now, role of innate immunity for acute rejection. Here, there is a role because the innate immune system has to facilitate the inflammation by complement activation, recruitment of leukocytes. There will be killing and there will be adaptive immunity which falls into place. So if there is an antigen and antibody reaction immediately after the transplant due to preformed antibodies in the system, the innate immunity will activate the cascade with the complement activation. So this is how the ligand and the damaged cells are going to act, that all the cells are going to recruit the natural killer cells, the cytokines, the inflammatory cells, and it is going to cause a destruction and enhanced killing of the donor cells. This is the classical complement pathway which is activated in the acute antibody mediated rejection. So there is a complement, there is an HLA antigen, and there is an antibody. So if you see this light blue structures, they are HLA antigens on the donor cells. You have an antibody from the donor, from the recipient serum, which is circulating. It will engage with the HLA antigens, and a complement will bind to these antigen antibody complexes and set forth a complement cascade. So if you look at the complement cascade, there will be the convertase enzymes, which is converting this to C2A and B. And then there is a C4, which is again differentiated, and C4D is deposited in the capillaries. Either the C3 convertase then leads to an accommodation reflux, or there is a destruction and it causes a enzyme cascade. Once you have the C5 convertase, there is a chemotaxis, there is inflammatory cell inflammation, and there will be a membrane attack complex formed, which is going to punch holes in the donor cells. So these holes are going to cause lysis of the cells. Now I have written eculizumab over here because this is the place where a eculizumab or an anti-complement factor antibody will act. And here you can stop when you know that there is an antibody mediated rejection and you give eculizumab, the complement is cut off and the reaction stops. So this is the evolution of an immune response after the kidney transplant. So first is a trauma of transplantation, that is an organ is removed, perfused and transplanted. There's expression of pro-inflammatory cytokines and recruitment of inflammatory cells such as macrophages into the allograft. There's presentation of the antigen which is coming with the donor to the recipient T cells, which then causes migration of the passenger leukocytes into the host lymphoid tissue, direct, indirect, and semi-direct antigen presentation, and entry of the recipient leukocytes into the transplant. Then there are activation signals for the T cells. So these signals are stimulation of cells by T cell receptor signals, co-stimulation and cytokines. So these steps I'm showing you because these are the various steps where the various immunosuppressants work. So steroids are going to work at the migration of the passenger leukocytes. IL-2 blockers are going to work at this level where you have the first signal and the second signal come in. Then there is a generation of different types of immunity where the innate immunity and then the adaptive cell-mediated and humoral immunity are coming in. Migration of activated leukocytes to the graft. There's upregulation of the MHC, which I will tell you a little later, and adhesion proteins on the graft by the cytokines like the interferon alpha and TNF alpha, which attract the leukocytes and the chemokines. And then there is a destruction of the donor tissue. Is ischemia reperfusion injury, which again has a production of or exposing the antibodies in the donor cells, and that is what causes more of the rejection. Sometimes there are autoantibodies products made because of exposure of these cells, and they can also sometimes initiate the cascade of rejection, and this comes into play in non-HLA antibody type of rejections. These are the different antigen presenting cells. There are dendritic cells, there are macrophages, and there are B cells. So dendritic cells, there are naive T cell activation and clonal expansion in differentiation into the effector cells. Macrophages, the effector T cell response is macrophage activation and cell mediated immunity. B cell, there is effector T cell response where B cell activation and antibody production takes place. So there is an antigen uptake, there is an antigen presentation, and then there is a response to it. Now the cellular component of the adaptive immunity. So you have antigen recognition. Say there is a B cell lymphocyte, there is antigen recognition, there is effector function, which is neutralization of the micro or the foreign element by phagocytosis. It ingests the phagocyte, but this doesn't come into play in transplantation. Helper T lymphocytes, here it is activation of cytokines, activation of macrophages, inflammation, and then proliferation and differentiation of T and B lymphocytes. There is cytotoxic lymphocytes, which affect the effector action is killing of the infected cells. And then there is Tregs. Tregs are very important in transplant because the moment Tregs come in, there's a suppression of the other lymphocytes and the transplant rejection cascade totally stops. So the key principles of immunology are self or non-self discrimination. This is very important because when a cell or a donor organ comes from a donor and it is not from the same identical twin, then it is non-self. So the ability of the body to distinguish between the body's own cell and the foreign invaders like bacteria, viruses, or transplant tissue, which is non-self, it is directly taken as an enemy and it has to be attacked. Then there is recognition, the capacity to recognize and mount a precise targeted response against a very vast array of unique foreign molecules, that is antigens. That is recognition which our immune system is able to do. And this recognition comes partly from innate immunity and partly from the adaptive where this being has already seen that antigen in the past and a memory response recognizes it. Immunological memory, so the ability to remember the previously encountered pathogens, allowing for a faster and a stronger and a more effective response upon re-exposure. So this is like re-challenging the immune system that something has already caused a reaction in the past, has already given a memory cell to the body, and the antibody antigens will be recognized and a severe response will take place. This is where vaccines come into picture, that we give vaccines, we induce an adaptive immunity to the microorganism which is given in the vaccine, and when the infection comes in, the vaccine which has been administered comes into effect and it tries to kill the bacteria corresponding to it or virus corresponding to it. Regulation and limiting response. This is very important because if the cascade continues to go on and on, then your own tissue or the body of the host is also destroyed. So the termination of the response is very important once the pathogen has been cleared and T regulatory cells come into action. So these principles ensure protection from the infection without causing autoimmunity or excessive damage. So these forms the basis of our understanding transplant. So what are the effectors of rejection? T cells, B cells, antigen presenting cells, and MHC. T cells arise from the thymus, from the bone marrow derived precursors. Each of the T cell has unique T cell receptor clone. Selection can be positive selection or negative selection. There can be CD4 uh, T cells which are antigen specific immune response, or CD8 T cells which are precursors to the MHCs. B cells will arise and mature in the bone marrow and they have a negative selection. They express B cell receptors on their surface. When this is stimulated, the B cells will secrete antibodies of the same specificity as the B cell receptors. So they can produce antibodies. So when you want to stop the production of antibodies, you have to suppress the B cells. Antigen presenting cells, these are very important. They activate the T cells, endocytize the antigen, then display it on the MHC molecules. T cells will recognize and interact with the antigen MHC to become activated. Now, what is the MHC complex? The major histocompatibility complex it is called. It encodes molecules crucial to the initiation and propagation of immune response. The HLA complex on the chromosome 6 contains over 200 genes, more than 40 of which encode for leukocyte antigens. The HLA genes that are involved in the immune response fall into two classes, class 1 and 2, which are structurally and functionally different, which I will show you in some time. So the HLA system is the genetic loci involved in the rejection of the foreign organs which are known as major histocompatibility complex and highly polymorphic cell surface molecules encoding the MHC. The human MHC is called the HLA or the human leukocyte antigen system because these antigens were first identified and characterized using the alloantibodies against leukocytes. So these were first diagnosed in pregnant females where they had human leukocyte antigens or hum-agglutinins it was called. And this was seen in previously transfused patients or pregnant women. Graft rejection was found to be associated with development of antibodies against these allogenic leukocytes. The HLA system has now well been known as transplantation antigen, but the primary biological role is regulation of immune response. So this is the chromosome 6 and the short arm of chromosome 6, that is this arm, has encoding for class 1, class 2 and class 3 genes. The more important ones for the transplant being class 1 and class 2. So types of MHC, there are three classes of MHC molecules, that is the HLA antigens. Class 1 encodes for glycoprotein expressed on the surface of nearly all nucleated cells. The major function of class 1 gene is presentation of the peptide antigens to the cytotoxic T cells. Class 2 encodes for glycoproteins expressed primarily on antigen presenting cells, example macrophages, dendritic cells and B cells, where they present processed antigenic peptides to the T helper cells. And class 3 encodes for various secreted proteins that have immune function, including components of the complement system, C2, C4, factor B, and TNF, and the molecules involved in inflammation. So this is how things are, that when you have nucleated, all the nucleated cells will have class 1 MHC, class 2 MHC are antigen presenting cells. RBCs hardly have any antigens only. So this also forms a basis for a clinical caveat, clinical pearl, that when you transfuse a patient who is going to go for a transplantation later on, you should give pure RBCs rather than a whole blood, because the whole blood will contain all nucleated cells and leukocytes and lymphocytes which are nucleated, and they will have class 1, class 2 MHC. But RBCs are devoid of these antigens, and if you give pure red cells, the patient will not get sensitized to HLA antibodies, which can later become a problem during transplantation. So the functions of both the class 1 and class 2 molecules is to present short pathogen-derived peptides to T cells, a process that initiates the adaptive immune response. Class 1 samples the cytosolic proteins and detect the foreign proteins that would indicate an intracellular pathogen, such as virus or intracellular bacteria, recognized by CD8 T cells and provide a surveillance mechanism to target the infected cells. Class 1 codes for the polypeptide gene of class 1 molecule. The beta chain of this is encoded by the gene on chromosome 15. And there are some 20 class 1 genes, but for us, the important ones are A, B, and C. These are the main actors in the immunological theater. So what is the structure of class 1? This is important because when we type the class 1, we are only typing for alpha 1, alpha chain, not beta chain, because beta chain is very small. There are four regions. There is the cytoplasmic region containing the sites for phosphorylation and binding to the cytoskeletal elements, and transmembrane region containing the hydrophobic amino acids. Class 2 encodes for alpha and beta polypeptide chains of the class 2 molecules. The designation of their loci on chromosome 6 consists of three letters. The first is D, which indicates the class. The second is M, O, P, Q, and R, and the family. And the third is A and B, the chain. So alpha and the beta chain. So here we are mainly looking at DP and DQ, alpha and beta, and DR, beta. Okay, so that is the chains we are looking at, and this is a typical diagram of the class 2 MHC molecule, which unlike the class 1 has two chains. There is an alpha chain and a beta chain. So when we are typing these, we want DR beta 1, 3, 4, 5. We want DQ alpha, DQ beta, DP alpha, DP beta. These are the typical class 2 antigens which we are looking at. So that was about the HLA. Now let me tell you about the antibodies, because we looked at the antigen. Now we are looking at the antibodies. I told you that when antibodies are formed or when there is a memory response, it causes a rejection. So what are these antibodies? These are donor specific antibodies which can be directly attacking the allograft if there is an allo antigen specific antibody in the recipient. So if you have a preformed DSA, then you have these because of pregnancies, blood product transfusions, or previous transplantations, and these will cause an increased risk of acute or chronic antibody mediated rejection at the time of transplantation if you have circulating donor specific antibodies. After the transplantation, if there are low-level preformed DSAs, they can cause an hyperacute rejection or an accelerated rejection, which I will tell you in some time. Or there can be a chronic antibody-mediated rejection if there is a low-titer antibody which is chronically damaging the allograft. Post-transplant, there can be formation of new donor-specific antibodies because of exposure of new HLA antigens to the system, and the host system is recognizing or identifying foreign antigens, processing them, and making them into new antibodies. This will not happen if the immunosuppression is adequate, and the de novo antibody formations will be suppressed. Now, what is hyperacute rejection? This takes place within 24 hours of transplantation. Serum antibodies react to foreign MHCs, triggering the complement system. The inrush of the neutrophils and the inflammation causes a clot formation in the blood vessels. The graft dies without being vascularized. So it is very important that if you have antibodies already in the system, like I showed you in this cartoon, then at the time of transplantation, they already have an antigen to which they can attach to in the donor allograft, because the blood goes in with the preformed donor specific antibodies. This will bind to the HLA antigens on the surface of the endothelial cells. It will form a clot, it will form a cascade, and it will not allow the graft to get vascularized. Acute rejection, which can happen within 24 hours or within the first month of transplantation. This is an old-fashioned immune response where within 2 weeks of transplantation, the macrophages, the lymphocytes swarm the tissue, triggering cytotoxicity, complement activation, and graft cell lysis. The 2-week delay is indicative of the T helper activation time. So it is a smoldering response wherein maybe a memory comes in that you have had this antigen exposure in the past due to transfusions or pregnancies or previous transplants. And once the allograft comes in with a similar antigen or the same antigen corresponding to those antibodies, it incites a response, and at 8 to 9 days, the antibody mediated rejection comes in because of the T helper cells and the antibodies being released. What is a chronic rejection? Even if a graft escapes an immediate rejection response, it can undergo rejection years later. Tissue typing and immunosuppressive drugs decrease the likelihood, but they have a long way to go. So if we reduce immunosuppression, we do not monitor for newly formed antibodies or old antibodies in the system and do not treat them, they will cause a chronic destruction of the allograft, which cannot be reversed with any of the strategies even present in today's modern era. So what are the zones of immunological privilege? Transplants into zones of immunological privilege have proven highly successful. For example, few blood vessels in the cornea, like these are privileged transplants. Cornea does not have blood vessels, so there's a very less chance of corneal graft rejection. So I have a patient who's following up after a second corneal transplant, and these are the situations where we give immunosuppression. In the first corneal transplants, we hardly give any immunosuppression because they are now immunogenic. There has been some success in transplanting fetal pig neural tissue into the brains of Parkinson disease patients. Here also there is not much rejection seen. Graft versus host disease. This is very important. Like I told before, that lymphocytes from the donor are carried by the organ into the body of the graft recipients via bone marrow or with the intestines. If the recipient is immunocompromised, the foreign lymphocytes can attack the host tissue, resulting in skin rashes, intestinal problem, organ failure, and even death. Liver, spleen, and bone marrow transplants all carry the risk of GVHD. What do we need today? We have now understood that what is the basis of a transplantation immune system which can attack the allograft. We now want to know how do we understand what immunosuppression to give, how do we understand what is the chances of rejection with this particular donor. So we need to identify the foreign antigens from the immunizer, that is the donor, and the pathogenic antibodies which may be present in the recipient. We need to immunostratify a patient to identify low, high risk patients and tailor the immunosuppression and avoid the risk of infections. Because if you over immunosuppress, they'll get infected. You under immunosuppress, you can get rejection. So if you know the perfect mismatch, if you know that the donor coming into this recipient is mismatched at XYZ antigens, or the patient has antibodies against this donor, you can perfectly modify the immunosuppression, not too much, not too less. Be able to transplant sensitized patient with optimal outcomes by suitably desensitizing them or giving an acceptably mismatched donor against whom they will not have any reactions, have the ability to monitor them post-transplant and prevent the graft damage. The first is HLA typing. We need to first understand that when a donor is giving a tissue to a recipient or a patient, how much different is it going to be from the patient? So you need to do HLA typing. That is, you are identifying the non-self. Class 1, A, B, and C loci. Class 2, DR, DP, and DQ. Like I told you, when MHC class 1 and class 2, we want ABC typing. Class 2, we want DR, DP, and DQ. Pre-transplant, these are important because mismatches in these are a matter of immunological risk. Mismatches also impact the short-term and the long-term outcomes of post-transplant recipients, and they are required when immune events happen to identify if there are antibodies against those non-self antigens which have come with the donor. So the HLA typing, whenever you read a report, it will have either a 2-digit or a 4-digit or an 8-digit, and this is how the nomenclature looks like when you look at a report. So if you have an HLA-A and there's an asterisk over there, which you can see, it means it is a molecular based typing. The first 2 digits specify the HLA protein. Okay, so that is the antigen. 02 is the antigen. Then the next comes the allele part of it. That is the 0101. Okay, and then later on the other part is required more in detail for bone marrow transplants because you need perfect matches. In kidney transplant, we can do with 4-digit typing because those 4 digits will be able to tell us how much is the mismatch and whether there are antibodies against these. So this is how the HLA typing will look like. And if you look at this HLA DR beta 1, DR beta 1 asterisk 02 means DR beta 1 is the gene and 02 is the antigen. DR beta 1 0201 means specific HLA protein or allele. So this is the allele 101, and you will have a coding region and a non-coding region coming after that, which is important only for the bone marrow and not for the kidney transplantation or solid organ transplantation. Now, this is a very old paper from 1990s, where it was elegantly showing that the more the match, the better you are. So if you look at this graph, you can see that your allograft outcomes are worse when you have complete mismatches. More than 6 mismatches, you have the most inferior outcome. You have good matches, more than 6 matches, you do the best. So 0 mismatches does the best. 1 mismatch after that, 2 mismatches. So if you see this list, it is showing that the lesser the foreign tissue you get, the better is your outcome. So when you do the HLA typing, you're going to do the matching. And when you see that the match is more than 6 matches or there is a 0 mismatch, it does the best. So that was the HLA typing part of it. Nowadays, it is done with NGS by the genetic technologies where you get the full string. You can do it on the Luminex platforms where you get 4 digits, which are necessary. But these are done for every transplant to understand the mismatches and the matches. Now we have looked at the antigens coming in. Now we want to look at the antibodies which are there in the patient's circulation, which are going to cause a problem at the time of transplant. So how do you identify these antibodies in the system? There are cell-based approaches where the donor lymphocytes are taken and they are incubated with the recipient serum, and the complement is added. You can either see the cell lysis on the under the microscope, or you can run it through a flow cytometer to see if the serum of the recipient has killed the donor cells which have been incubated with it with the complement, which is what happens in your system. So it's like a mini transplant done in the lab. These are done immediately before the transplant or at starting the workup to understand if this recipient is going to reject the donor transplant kidney coming in. Next is the solid phase assays. These are the more advanced assays where donor cells are not required. You are only using the serum of the recipient and you are running them along with inert beads through a Luminex platform which are coated with antigens. So imagine a donor cell which has antigens attached to it, and you are incubating them with recipient cells and adding complement to mimic the immune system and trying to see under the microscope if the cells are killed like it would be in the body. Now you're doing it solely ex vivo, and you are trying to see that there are solid phase assays where no cells of the donor are used, but there are beads which are mimicking the donor cells, and antigens which are known to us are attached to it. The serum of the recipient is incubated with these beads, and then they are run through a Luminex platform, and the lasers will pick up if there were antibodies with attached to the beads with a named antigen on it. And that identifies the antibody on the bead. So these are the different types of antibody identification which I just told you. This is the CDC, that is a complement dependent cytotoxicity crossmatch, where a donor cell is there and it is incubated with the serum of a recipient which might have antibodies. If there is a corresponding antigen on this donor cell, like it is shown here, these antibodies will attach to that antigen. Then a complement is added, and then you can see if the cell is lysed or no. This can be AG enhanced, and this can also be done via flow cytometry assay, which is a which is an objective assay, which will tell us how much of antibodies are there. Then comes the solid phase, where I told you the cells are replaced by inert beads where known antigens are there. So I will know that which bead has which antigens. And if that bead is flagging positive, I know that the patient's serum has these XYZ antibodies on it. So it is a very precise system to identify the antibodies. Because there were drawbacks of the cell-based assays where low antibody titers are not being picked up, and sometimes the cells may express these antibodies in a low level. There were instances where rejections were happening when only CDC crossmatch or flow crossmatches were being used, and hence the solid phase assays have come into being. These are the solid phase assays which I showed you, that there are inert beads where there are antigens attached to the beads. One is a screening bead where class 1 beads are different and class 2 beads are different. The serum running through around the beads will have antibodies, and if there is a corresponding antibody, it will attach. It will give you only a yes or a no. It can give you a percentage of positivity of the beads. If you want to identify which antibody, you have to use single antigen beads where a sphere or an inert bead is attached with only one type of antigen, for example, A2, A24, B27. So these will have only a single bead attached. So when that bead is attached with the antibody and it is running through the Luminex system or the machine, the laser will pick up these antigen antibody reaction. It will tell us which antibody is there. So the investigations to be done pre-transplant, one is HLA typing, which you want to do because you want to identify what is not matched or what is foreign. You want to do cross-matching. You want to identify if there are antibodies present against that particular donor. And you might want to do a solid phase assay like a single antigen bead assay if there are antibodies present in the patient's serum which are detected on the screening platform. So an HLA lab becomes the basis of your risk assessment. So we have come a long way from cell-based assays. This is how the CDC cross-match seems. And now we are doing bead-based assays. So in the pre-transplant risk assessment, we want to do the donor and recipient HLA typing, which tells me the risk between this donor and recipient. I want to do HLA antibody assessment as to what antibodies are there in this recipient's serum, which will tell me that no donor is going to suit him. At the transplant, I'm going to do cross matches, CDC cross match and a flow cross match, which will tell me whether this kidney will immediately reject or no. Like I showed you in hyperacute rejection, that you already have some antibodies formed, the kidney is coming in, and these antibodies attack it. So that can be picked up by the cross matches done immediately before the transplant, and you can prevent this phenomenon. After the transplant, you have a risk of memory response for which the HLA antibody monitoring with these solid phase assays help. And they also help you in diagnostic antibody mediated rejections where the biopsy is done and the serum is sent to identify if there are antibodies against that particular donor who had given the kidney. And you can monitor these antibodies post-transplant. So now let me show you this case. 37-year-old female suffering from CKD and chronic GN, working up for living donor preemptive renal transplant, donor being her mother. She has history of blood transfusions once in 2012 and has history of 3 pregnancies. So now in this story, I have told you that this is a young female. She's being worked up for a living donor transplant. Donor is her mother. So being her mother, I'm sure there'll be a half match at least. She has a history of blood transfusion and pregnancies, which means that her body might have developed adaptive immunity to the HLA antigens which came from the 3 pregnancies and from the blood transfusions which came from other human beings. So this patient is called sensitized. When this patient is sensitized and you're going for transplant, you should be wary that these antibodies can come and attack the allograft if they are present in the system at the time of transplant, and or a memory response can happen after the transplant because they are stored in the memory cells. So when I want to work up this patient, one, I want to see how much is the match with her mother. So if you see the recipient's HLA, you see a 0301, which is a match with her mother. All the ones in blue are a match. All the ones in black are a mismatch. Okay. So HLA A, B, C, DR beta 1, DP. You see a DP alpha, DP beta. So like I had explained to you, in A, B, and C, you have alpha change. So you don't need to type 2 things. But in class 2, you have an alpha and a beta change. So you need both these to be typed to understand what is different. So when we were working up this patient, we have done the HLA typing with a molecular technology, and we have seen that the HLA match at these 11 loci are 9 by 16. The CDC cross-match, that is a complement-dependent cytostatic cross-match, auto T cell and B cell are negative, allo T cell and B cell are negative, which means that the recipient serum did not react with the donor cells and there was no lysis. So this was a negative cross-match. Now, should we stop here? Because many places even in India, in some places, they stop at CDC cross match. But if you see a sensitized situation and you see this and you go for transplant, suppose there is an antibody to any of these mismatched antigen, because finally, whatever is matched. You're not going to have a rejection to that. But whatever is mismatched, if there is an antibody to these mismatched antigens, which she has got through the sensitizing event past, like transfusions or pregnancies, she might still reject. So we should not stop at the CDC. Okay, so we should look further. So we did a PRA screen. That means we have screened her recipient, the serum, for any antibodies if they are present with the screening beads. Which screening beads? I will show you again. These are the screening beads. So this screening bead will only tell me yes or no. So when I screened her, I have seen that her class 1 was negative. So in class 1, she has no antibodies to any HLA antigens. But in class 2, she had antibodies, so it flagged positive. Mind you, this is a screening mode. So we are not going to get the identification of what is positive. But it is just telling me that class 2 antibodies are there in her serum. Now I want to know which antibodies are there and are they against her mother. So this is the single antigen bead assay. It looks like this, that you have the name of the antigen, you have the allele, and you have how much antibody this patient has. So that is the strength of reactivity of that bead. Okay, so now this is the list I got from the single antigen bead to identify which antibodies are there. Now I need to see whether any of these correspond to the mother's mismatched class 2 antigens. So let us see. So here 1502 turned out to be a donor specific antibody, and DQ antibody, DQ alpha beta 0103 and 0601 turned out to be donor specific. So if you look at this DR15, look at this DR15. The MFI is 13801, which means that this is a very strong antibody that this patient has in her serum, which could attack the donor cells as soon as the transplant happens or in some time afterwards. The crossmatch did not pick it up because the levels of these antibodies are lower in the threshold than what a crossmatch can pick up. Okay, so here the summary of the case is that mother to daughter with more than one haplotype match, CDC cross match is negative, HLA mismatch at A, B, C, DR, DP, and DQ is 9/16. HLA match is 9/16. So in any situation, anyone would think this is a low risk transplant and go ahead. But no, this could cause a problem because there is a donor specific antibody to DR beta 1 and 15 and DQ. Later on, a flow cross match was done, which showed that the B cell was positive. Now remember, B cells have class 2 HLA antigens and it turned positive. So the CDC crossmatch will pick up big amount of antibodies. Flow crossmatch will pick up moderate amount of antibodies, and the solid phase assays will pick up even low levels of antibodies. So when I'm evaluating a patient for transplant, I need to know what are the mismatches. I need to know whether there are any antibodies to these mismatches and then plan the transplant. So this is how we make an algorithm, which we have now published in literature, that at solid organ transplantation, whether it is living donor or deceased donor, you should have a complete typing of intermediate resolution at A, B, C, DR, DP, DQ, that is molecular typing. And you should have a screening which tells you whether this patient has antibodies, yes or no. So if the screening is negative, then you keep screening every 3 months till the transplant is done. At transplant, you do the cross matches, like I have told you, cell-based cross matches. If they are negative, you go ahead for transplant. If anything turns positive, then you turn to doing single antigen bead, which will tell you which antibodies are there. Identify those antibodies. You may abandon the transplant or you may want to desensitize and go ahead. So a lab and a clinical correlation is very important when you are trying to do a transplant, and that is where I will leave you today. Thank you.
Thank you, Dr. Shruti. I think that was quite an enlightening presentation. I just liked the way you started with the milestones. I think from where we started, I think in the 80s or even way before that, and where we are currently. I think at some point, yes, you reminded me a little bit on the immunology class, but everything, I think you brought everything home with the case at the end, which was quite good, just to summarize everything up. So thank you for that. I think I'll just want to open up the session to any questions, any reactions to the discussion.
Can you differentiate for us between DSA screening, pre-transplant, and PRA? Yeah, so DSA, there is nothing known as screening. DSA is identification of donor-specific antibodies. You can't screen for it. You can screen for presence of HLA antibodies by doing a panel reactive screen. That is known as mixed screen. Like I showed you those beads which have multiple antigens on it. It can tell you yes or no. Once you have a yes or no, you want to identify a DSA for which you need a single antigen bead assay. Okay, so I'm typing there SAB to identify DSA, PRA to screen for HLA antibodies. That is how it will go.
Thank you, Dr. Shruti. I think we have Professor Bima. His hand is up. Prof. Bima.
Thank you very much, Yuri, and compliments of the new year to everyone. Dr. Shruti, thank you very much for that wonderful and enlightening talk. My question is, often when we do transplants and the patient has low MFIs, we still go ahead and transplant the patient. What is your cutoff?
So every lab will have its own cutoff. We keep a cutoff 1200 to 1500, after which we call something positive. But
Yes.
if there are strong antigenic antigens like DR beta 1 or B35 or A24, these are very strong antigens. If they are to the tune of 2000 or 3000, we will still take cognizance and give plasma exchange and desensitize and take it ahead. If they are 1500, 1600s, we will neglect them. Cross matches, of course, will come negative with lower than 5000 MFIs. Below 15,000, the CDC will come negative, and below 10,000, the flow will also come negative. So when they are negative, there is a good literature, there's a good paper from Dr. Dechu, from Dr. Stan Jordan's group, where, they've done a risk stratification where you add up the MFIs of whatever is positive as a donor-specific antibody, not all HLA antibodies, only donor-specific antibodies. So you may have low MFIs for all HLA antibodies. That doesn't mean anything. If there is a donor-specific antibody which is positive, and say A1 is 5,000, say B35 is 2,000, so you make it 7,000. So there is a risk stratification score. So if the score goes to more than 17, they abandon the transplant. If the score is less than 7, then they will go ahead with the transplant with all the desensitization protocols. So it is the cumulative score of the HLA and not the individual ones.
Well, thanks. That's very helpful. Because often, in the panel meetings, people argue that the MFI is like 10,000 something. Say, let's go ahead with the transplant. But, we are not really stratifying it. So I think we need to do
No,
that.
that's what you
Yeah.
have to understand which
Yeah,
antibodies these are. You cannot just say that these will not do anything.
sure.
You have
Well,
to
thanks a lot.
identify them as a DSA or non-DSA. See, there is a test available in market which is dirt cheap, which is known as DSA crossmatch, but it is rubbish. You do that and you get misled. So you should never use that test because it will not identify the antibody to you, and it can be false positive and at times false negative. So if you want to identify the HLA antibodies against the donor, you need a single antigen bead assay, because that will tell you which antibody is there against the donor to which quantity.
Okay, great. Thank you very much. That's very helpful.
Thank you, Shruti. The wonderful presentation. I mean, it shows the
I
mastery.
hope it was not complicated. I was feeling in between that I've made it complicated. I was trying to be as low as possible.
It shows
It was a
mastery.
master class in
Yeah,
immunology.
masterclass. Yeah. So I mean, one quick one. No, still after all this testing, you can be caught by surprise having an antibody which is missed. Do you?
Yes.
How do you sort of then? What do you do at that point in time? And you have a rejection still. Then what do you actually look at, and what do you do then?
So we look at the pathology. So see, there are various caveats of these Luminex systems.
Yeah.
These single antigen bead assays may not cover all the population HLA antigens. Because say African population, Indian population, these are very ethnically diverse populations. Unlike the Caucasian population where you will have only few antigens coming up again and again.
Okay.
So you have Asian beads available and you have Caucasian beads available. So we have actually tried to see how many antigens and alleles are not represented on these panels. And I have just published a paper in IJT where 25% of alleles are not present in either of the vendor beads. Antigens are present, but alleles are not present. So what I try to do is when I report something, I try to do the SAB and write in the bottom that XYZ antigen allele is not represented. So even if the virtual cross match is negative, please do the flow cross match before the transplant, because that can help you pick up some antigen which is not represented. That is one big caveat. Second caveat is that today if I do a single antigen bead assay and the patient is in a quiescent stage, you may not pick up any antibody. So you might want to do some single antigen beads at varied intervals like 6 monthly, 8 monthly, till the patient gets transplanted to get a cumulative image of the patient. Because when you look at a serum, once it is a snapshot, you may never know what antibodies were there in the patient when the patient never met you before. So that is a reason why you may sometimes get a rejection from a memory response of something the patient got long back, which was not in circulation when you did the test. So these are the situations where you will get a rejection. So when they are sensitized patients, like people had pregnancies, people had transfusions, I like to repeat the single antigen bead multiple times to get a idea of circulating antigens, especially during a viral infection or something like that, because all those antibodies do come out in the system at that time. So that is one thing. And last is that sometimes you may still not understand why the rejection happened. Sometimes non-HLA antibodies also come positive, like AT1 receptors and AECA. Like I showed you that during an ischemia reperfusion injury, you may get auto non-HLA antibodies which are formed to the endothelial cells or the AT1 receptors. So in rare situation, 4 to 5% of the situation, you may have non-HLA related ABMRs, which are very difficult to reverse because they come with TMAs.
TMA, okay, right. Oh, that is very scary actually. This is the only scary part. You do everything, your serum is negative, and suddenly now your creatinine is increasing immediately post-transplant or sometimes
But
on the
I think we have to attack immediately. So keep a very low threshold for a biopsy and do a DSA at the same time and do non-HLA antibodies. And if the biopsy phenotype is that of a rejection, then cellular, then of course cellular. But if it is an ABMR, then initiate plasma exchange and IVIG, because an ABMR treated early will reverse back. We usually do IHC stainings also with CD3 and CD20 so that I know whether it is a T cell mediated, B cell mediated, and I know whether ATG or Ritux has to be given.
Okay, right. And is this like in 100 transplants, how many would you find coming up like this, like with a rejection that is still after all the testing is negative? And
2
some
to 3%. 2 to 3%. Yeah.
Right. And now these antigens, is there any way that actually you can classify, you can actually characterize these antigens and put it into like an Asian SAB or African SAB? Is there any way that it can be done, like
So
working with the lab?
the companies do have Asian beads. So like I said, if I pick up Caucasian beads, then I'll have more discrepancies. But what we are using are Asian beads, and as we speak, the companies are adding the antigens which are being which are being identified as the population goes. So even Africa, we will have to see when if we have HLA typing of say a population, then I can see from both the vendors what is absent and what is present.
Okay. But
So
this
that
character, yeah, go ahead. Please go ahead. Yeah.
So these antigens, so we have to see what is different in the Caucasian population, Indian population, and the African population, because China, Japan, so Asia, they have similar kind of beads because they are similar kinds of HLA antigens. In India, South India, tribal areas, West, North, they have a different, they do have different HLAs. Which, so in South India, they found 35% discrepancy between the what is represented and what is not there. We
Wow.
found 25%, but that 35% study was some 5 years ago. So after that, the companies have added a lot of antigens on it. So I would say that whichever lab does the single antigen bead profile for you has to do that study. So we actually gathered a lot of HLA data and tried to see what is present and what is not present.
Okay. Right. Bioinformatics, does it help in any way identifying these sort of antigens, or are there any sort of things?
Which repeat your question, I didn't get it.
No, like use of bioinformatics, identifying these.
Bioinformatics.
Yeah.
No, I think it will only have to have HLA study of the population.
Okay, right.
So
Okay.
if you have a city or if you have a region and you have the HLA from the bone marrow registry,
Okay, right.
then
Okay.
that bone marrow registry has a lot of donors. So that tells you what type of HLA antigens are prevalent in that population.
Okay, right.
And then we can see the single antigen bead profiles available there, whether they are there. So that is the best way to get to know what antigens are there and what are not there.
That's a great idea. Yeah, that's wonderful. Okay. Okay. So then actually
Yes.
these are there's a nomenclature for all these antigens that we pick up on this sort of the bone marrow
Yes.
storage.
Yes.
Okay,
This is
they have.
the like the nomenclature I showed you. Same way. There will be A, B, C in the front. There'll be a molecular asterisk sign. There'll be an antigen. Then there'll be an allele. Then there'll be a coding region, non-coding region. It will be in this manner.
Okay. So then these that you identified from the Asian side or from the Indian side, now you have identified and you have them all in the nomenclature now, which is not there in the Western view.
They are there in the nomenclature.
No, they are there in the nomenclature.
But they are not represent. Yeah, but they are not there on the beads which are being provided by these vendors to identify antibodies. So suppose there is a donor coming with an antigen which is not there on the bead.
Yeah, no, yeah, correct.
I will not know if that antibody is present, no?
Correct, yeah, absolutely.
And the patient may reject.
Yeah. So that is very scary. That's the only scary part in transplant.
So that's why every time we type the donor, we type and keep the donor, and then we look at the bead profile whether that antigen is there on that bead.
Okay.
If it is not there, we write a caveat below in the report that note the bead does not have the donor representation.
Okay.
So the clinician is alerted that you may find a rejection if there is an antibody to this, which we have not been able to pick up. And a flow cross match then becomes very important because the flow is very sensitive. So if that is negative, then your first 10 days are sorted out anyways. Then if the lower level of antibodies are there, then they will cause rejection later on.
Later on, yeah. Okay.
But flow will at least take you through for the first month.
Okay, first month. Yeah, that's at least something. Yeah, very good.
Yeah, because once it is there, then we can simply say that, okay, this was an antibody which was not, this is an antigen which was not there on the bead, and I have to beware if any antibodies form to it.
Yeah, there's a lot of, see, on the African side, I think there are a lot of challenges. The laboratory, creatinine variability, the internal, external validities of the, the lab, creatinine, then the blood levels of, the drug levels. These are all major challenges. But again, so coming again to another topic. Now, you run a big a very nice short program in Mumbai, So how
Yeah.
often do you run it? And, can you just give us some detail? Because there are a lot of people who are interested and actually would benefit a
So,
lot. Yeah.
yeah, so now in April, we are going to do a workshop. So that will
Yeah.
be like a conference which will be
Uh,
cornered. If I can just share the screen 1 minute.
yeah. Yeah. Wonderful. Yeah. Thank you.
Second, I closed it.
I think we put it into NASOT, isn't it, Damar? Yesterday I saw it. I took it from the Toronto group and I put it. I think we have shared it into NASOT, if I'm not mistaken.
Now, if you can see the screen, this is the Transplant Immunology Simplified and Solid Organ Transplant Infections 2026, where there'll be a full day workshop, which will be troubleshooting, and British Transplant Societies joining hands with us in this time. So 11th and 12th
visible.
Yet to
April.
load. Sorry.
It's not seen. It's not seen. Your
Yeah.
slide
It's
is
not
not visible.
seen?
No.
Okay, hold on.
Yeah, just try and reshare.
Okay, hold on. Let me just
Yeah.
share again. Share screen or window. Now?
Not yet.
So
It
it's
shows it's
loading and
loading
then it
something though.
disappears. Yeah. It
Not
loads
seen?
for a moment and then disappears. Yeah.
Oh, now?
Not yet.
Still not.
Oh, I don't know how it is, because this was the same present. Anyway, I will send it
Okay.
across to your
Yeah.
to y'all.
Okay.
And it is on 11th and 12th of April
Okay.
this year. It's a Saturday, Sunday. It is
Yeah.
a comprehensive way of dealing with day-to-day challenges in transplant.
Yeah. This is the Transplant Immunology Simplified and Transplant Infections 2026.
Yes,
That's the one.
yes.
Renal
So
Science.
on Sunday, on 12th of April, we have a full day of transplant work, immunology workshop, where
Okay.
cross matches, HLA typing, antibodies, applets, everything will be discussed, and there will be case presentations in the post-lunch session, and there'll be a quiz, or like rapid shooting of cases,
Yeah.
that what do you think, why this is like that, why this is like that, sort of a thing. And Saturday we will start by 10 a.m. with immunobiology of transplant. Then we'll be talking about rejections, pathology of rejections, chronic rejections, acute rejections, sensitized patients, how do you deal with it. Then the later part of the day on Saturday from 4 p.m. to 8 p.m. will be infections, where urinary tract infections, BKV, then pneumonias in transplant, diarrhea in transplant, approach to all will be discussed. We are just freezing the program in 1 or 2 days, and we'll put out the complete brochure also. Meanwhile, so I will send it to you also, specifically.
Thank you. I'll
So if
send it out.
someone wants to come, so these 2 days can be a very comprehensive thing.
Yeah. I'm sure. Very good, actually. Excellent.
Yeah.
Thank you.
Okay.
Yeah, thank you, Dr. Shruti. I'm not seeing any, we don't have any more questions on the chat box. So I think we are 24 minutes past the hour. So we'll close the session here. So I'll just give my vote of thanks. Dr. Shruti, thank you for making time. Thank you for taking us through this enlightening topic. And hopefully we'll have you back to again take us through more interesting topic on transplant immunology or any other transplant related topics. So on behalf of Africa Healthcare Network and our panelists, I want to wish you a good evening or good morning wherever you are, and till next time.